So one success to report from yesterday, which we'll show after lunch, is we had a videographer, Amber Coe, who's starting a new production company here in Boston, and she was there for about 6 hours doing interviews and taking videos of GitLabs, and she worked all night to edit the video into about a 3.5-minute presentation showing what GitLabs is all about, and so we'll show that when everybody gets back from lunch. So it's really good. It's amazing.
So before I introduce the next speaker, as PGP4, being in the PGP, as you all know, means having no shame. So I just wanted to mention that together with Nita Farahani, who Robert Greene mentioned in his talk, we've started a new master's program in bioethics and science policy at Duke. Application deadline May 31. And be happy to talk to you about it. And now if I can find Ting's presentation. Ting Wu is professor of genetics at Harvard University. At Harvard Medical School, her interests center on how chromosome behavior influences inheritance, and to this end, her laboratory invents and applies technologies that reveal how chromosomes interact with each other. Her talk will focus on a remarkable and puzzling set of sequences called ultra-conserved elements, which, like certain federal agencies, have resisted change for hundreds of millions of years. She recently co-hosted a symposium on genetics, biomedicine, and the human experience in space, and will bring some of that flavor to her talk. She is also the director of the Personal Genetics Education Project, pged.org. PGED works to accelerate public awareness of the benefits as well as implications of learning the details of one's genome, engaging the public through schools, conferences, the entertainment industry, museums, and a mobile game, which is at map-ed.org. She received her BA and PhD from Harvard University and is a recent awardee of an NIH Director's Pioneer Award. Her talk is entitled A Genomic Puzzle, a Potential Therapy, and Space.
I wanted to start off by saying it's a real privilege to be here in this room with all of you. And I was going to say in your genomes, but now I'm going to say and your connectomes. It really is a thrill to be here. Now, I'm going to talk to you about a remarkable set of sequences that are numbering in the hundreds to thousands, depending on how you define them, and are scattered throughout your genome, both in the coding and non-coding regions. Why are they remarkable? Well, they're ultra-conserved, as Misha mentioned, and we call them UCEs for short, first defined by Gil Bejarano and David Hausler and their colleagues. They are considered one of the most mysterious aspects of your genome to come from the genome era. What is it about them that is so extraordinary? Well, what it is they have not changed, as Misha mentioned, for 300 to 500 million years. That means they extend back unaltered to the time when mammals, reptiles, and birds first diverged. What does this mean? This means like, unlike most sequences in your genome, which have acquired and accumulated and maintained mutations through time, UCEs acquired mutations for a while, and then they froze, starting at about the point where that arrow is at. Why this happens, we don't know. What do they do, we don't know, but we think what they do might have something to do with the fact that they don't change. There are many explanations, and ours is shown here. I should tell you that this is a very speculative talk. Our model could be wrong, but we like it. It's not the prevailing one out there, and so I'd like to share it with you anyway. Our thought is that these ultra-conserved elements take advantage of the fact that your genome consists of two copies, one from your mother and one from your father. And we think the reason these sequences can maintain their sequence through all these years better than any other sequences that every once in a while they pair up and they compare the maternal and paternal copies, such that if something is wrong with one of the copies, the individual carrying that error suffers loss of fitness through disease or through death. Now, what that means is that when you have a genome where every UCE has a perfect partner, it can persist. But if that perfect partner cannot be found for all the UCEs, then what happens is you have loss of those genomes, leading eventually to the persistence of just genomes that have maintained those sequences perfectly. It actually explains ultraconservation. In fact, it drives ultraconservation. Now, how can we test this idea? Well, we thought we'd take advantage of the fact that your genomes are hugely dynamic. They are changing all the time. Someone mentioned somatic mutations, and that's what I'm thinking of when I say that. And the prediction is that if these UCEs do not like to be changed, and if they change, they lead to loss of fitness, then one might predict that these changes in your genome that are happening, that are good for you, or at least not harmful, should avoid the locations where these UCEs are in your genome. This test actually has not been difficult to do because people, through genome sequencing, have been identifying these various rearrangements that happen to your chromosomes. For example, some chromosomes lose material, leading to a shorter chromosome. Other chromosomes will duplicate material, leading to a longer chromosome. Some chromosomes will take a segment, excise it. Whoops. I hope you saw it. Excise it, turn it around, and reinsert it. And then some rearrangements involve bringing together two different chromosomes, having them exchange material, giving rise to two hybrid chromosomes. All right. How often does this happen? Extremely often. In fact, when Randy Buckner was talking to you about the connectome, I was thinking about how recent studies have shown that in certain segments of your brain, they believe up to 10 or more, 20, from your skin, 2% of your cells in those tissues will have acquired one of these changes. In fact, if you were to take a segment of a chromosome and superimpose on that segment the breakpoints of these rearrangements from 10, 20, 30, 1,000, 5,000 people, you would see that the majority of your genome is subject to these kinds of breaks. But not all of your genome. Now, where do UCEs lie? Some of you may have already guessed. Where they go is generally, not perfectly, but generally to regions of the genome that seem less subject to these breakpoints and to these changes. Interestingly, if you compare the location of these breakpoints between those representing healthy people from those representing people who are diseased, such as bearing cancer, what you'll find is that the diseased, the rearrangements that are associated with disease tend to be located a little more closely to where the UCEs are. All right. How do these breakpoints happen? Well, as you develop, your genome undergoes a lot of rounds of replication, and that replication process is not error-free. So many of these rearrangements happen simply by the process of development. Some of these rearrangements come from environmental onslaught. Some of these rearrangements, and I think because we are finding so many of them, by we I mean hundreds and thousands of researchers, because we're finding so many of them, I believe geneticists are beginning to really consider seriously the possibility or the probability that a healthy genome is one that is rearranging itself a lot. All right then, Misha mentioned space. One of the strong and persistent source of breakpoints for your genome are due to radiation from the sun and from elsewhere in the universe. That radiation is lowest for humans that live on the surface of the Earth. For those who live at higher altitude, that exposure to radiation can be up tenfold or so. And for those who will be or are contemplating interplanetary travel, that onslaught of radiation can be 100 to 1,000 times what a person normally experiences in a lifetime. So it's a big issue. Now, what my laboratory did is look at this interesting relationship between breakpoints of healthy people, breakpoints of people who have had suffered from certain kinds of diseases, and the location of UCEs. And we are wondering whether UCEs are an endogenous system of our genome to help protect that genome from rearrangements that are deleterious while still allowing the rearrangements that are beneficial. All right. up until recently, my laboratory has considered this ultra-conservation process as one that takes millions of years or hundreds of human generations. But as we saw these rearrangements appearing, and through our studies, we began to wonder if, in fact, the process of UCEs and how it protects our genome might be much faster. The idea is that these UCEs somehow can detect a mistake, a rearrangement in the genome that's deleterious, and get rid of the cell that carries that deleterious rearrangement. And so what we did is to select a specific set of studies, actually quite a few, that were done in a way where the rearrangements that were detected would be highly enriched for very, very recent ones. In fact, ones that probably occur in a person's lifetime. We even looked in cell culture, and what we found is that this culling away of cells that have deleterious rearrangements from cells that seem not to have those deleterious rearrangements does not take hundreds of millions of years. In fact, we think it takes simple cell generations, certainly shorter than a person's lifetime. And this led us to the beginning to think that it may be possible to harness ultra-conserved elements and have them work at our command to protect our genomes even more than they already protect them. Now remember, this is a very speculative talk. We have a lot of data, which I'm not showing you, but I am projecting where we think this research might go if we're correct. And we are fully prepared to be found incorrect. But anyway, let's see where we go. How might UCs work? Well, remember that pairing model I presented earlier. What we think happens is that your genome, every once in a while, or maybe every cell generation, pairs the two chromosomes you have for each, the two homologous chromosomes you have, one from your mother, one from your father. Now imagine what happens when that occurs for a chromosome that had suffered a deletion. What happens is that there will be a buckling. The same thing will happen for a chromosome that has had a duplication. We would anticipate a buckling. For chromosomes that have an inversion, we would anticipate an open buckle. And for chromosomes that actually consist of two hybrid chromosomes, we would anticipate a very fancy kind of buckle. So if that's what happens, then it may be possible for your genome to detect through UCEs changes that are affecting UCEs and are deleterious by our statistical analysis. So our idea is, in a normal, we're not talking about intervention right now, as you were growing, we imagine that at a reasonable rate, low but significant rate, your body is, has, and will be generating deleterious rearrangements. But if your UCE mechanism is working correctly, it will detect those rearrangements and destroy that cell. So as your body continues to grow, as you live, you will have an enhanced chance of having tissues that are free from deleterious rearrangements. Now let's talk about cancer. We've studied a lot of cancer genomes, and that's where we found association between cancer breakpoints coming near UCEs, which is not what you want. We wonder if in cancer genomes what's wrong is that the UCE mechanism has not been activated properly. What would happen in that case? Well, we think a cell that arises that has a bad rearrangement would not be culled and would therefore be allowed to grow. If that happens, then this might explain some of the tumors that arise. So what is our goal? Our goal is to understand how UCEs are working. Of course, we'll be testing our model. And if we can harness this mechanism, then we hope it will be possible to take tissues, individuals, organs that have a smattering or too many cells with bad rearrangements, and then cull that tissue, cull those cells of those with deleterious rearrangements. This treatment would be, interestingly, would potentially not require any diagnostics at all, because it's focusing on the structural rearrangement and not on the way on downstream physiological effects. In fact, if one regularly culled one's body of deleterious rearrangements, certain diseases may not even appear. Now I'm being fanciful, but I'm in the section called Explorers, right? Right. Now, this treatment could be general, and it would be efficient, and most importantly, because we're using a system that we think is already in your genome, might already be in your genome, it would be an all-natural process. And we are thinking, for those who are interested in working and playing off the planet, we are thinking that a system such as this might work well for those who are interested in long-distance interplanetary travel. And with that, because you wouldn't need a doctor, you may not need to have a lot of the clinical support that we need to have on Earth right now. All right then, so there's a genome we're trying to protect. And here is my lab. I'm a very, very fortunate PI. I have a wonderful lab. I'd like to thank in particular the individuals in red, Ruth and Chamath, did a lot of the recent work. Adnan Durti and Charleston Chang, who are no longer in the lab, did the initial observations of the relationship between UCEs and breakpoints in the genome. And we've had tremendous help from collaborators and colleagues, Amnon, Mike, James, and Steve, and wonderful support from NIH, from the Cox Fund, and from HMS. So I think I'll stop there. I'm happy to take questions.
Sure. So it sounds like in order to have this function, sorry, I'm Mary Wall. In order to have this function, you not only need to have the sequences conserved, but also you would expect that they would be maintained synthetically. Do you see that across the vertebrates?
You're asking about synteny.
Yeah.
So when studies have been done, these are not my studies, but studies done in a number of other laboratories, these sequences do seem generally to maintain synteny. And interestingly, they have some of them cluster. And the clustering pattern also seems to be somewhat maintained. Esther.
Esther Dyson, just briefly, are you working with NASA? Have you asked to genotype the astronauts? Anything that practical?
So we haven't, but the Space Symposium we ran was our first step to try and make that connection with NASA. Yes.
Because exercise tears down the system and it must be built up. Could this be a factor in eliminating the destructive?
Oh, interesting. So you're talking about physical stress to one's body could be damaging. Actually, there are plenty of studies saying inflammation is damaging. And one of the damages can easily be DNA damage. So yes, we would anticipate that if there's any truth to what we're saying, we would hope that it would be able to help in that situation. Thank you for asking that.
Hi, I'm Dan Turner, and I have a question. Would you think that this is in conjunction with things like autoimmunology, like with T-cell destruction, this correlation with apoptosis? And also, do you think there's corollaries to like poly-A tail erosion and to some of the major frameshift mutations or even chromosomal abnormalities?
I want to say yes to all of that. So thank you. The body has many, many ways in which it expresses disease, loss of fitness, illness, and many ways in which it fights off invasions that we think of as, well, we think about fevers and illness. So one of the questions, I can talk to you in more detail afterwards, but when I say that I think a cell or an individual suffers loss of fitness, we sometimes wonder if that loss of fitness actually is possibly caused by UCEs triggering a response to a damaged chromosome.
So, James Turner, parent of Daniel Turner.
Nice to meet you.
And not nearly as well educated. So I have kind of two quick questions. One is, I had always understood the conventional wisdom is that well-conserved parts of the genome are well-conserved because they're so critical to function that, a change to it would be that they're very close to the evolutionary kind of perfection model where just about any change to it is going to be deleterious. And I was curious how that fits into that. And the other question I had is, have you tried the approach of maybe trying to do some knockouts inside of UCEs and test the hypothesis that making some change in there is in fact kind of a death sentence for the cell?
Thank you. So let me answer your second question first. So yes, there are some technologies that have been emerging in the past 18 months especially. George's lab is responsible for, is one of the labs responsible for that introduction of a way to engineer genomes, and that has made it possible for us to start thinking about going in and knocking these sequences out to see if there's some effect. And now I've forgotten your first question. Oh, right. Yes, so conservation has generally, and I think is still, considered a very good mark for an important sequence. And yes, important sequences, especially those encoding regulatory regions and proteins and structural aspects of the genome, are extremely well conserved. These sequences are even more conserved than those. And I probably should have mentioned that there is really no obvious explanation that we know of, no molecular mechanism that can explain this level of conservation. So thank you for asking that.
Hi, Nathan Bowen, Clark Atlanta University and PGP 46. I really enjoyed that. Thanks for sharing. So this idea of homologous chromosomes pairing, right, is very variable across species, across evolution. Drosophila, I guess, their homologs pair, if I remember correctly, and you're talking about pairing at interphase, I guess, right? But
yes.
During interphase, I mean, not interphase. Yeah. So what is known about what causes homologous pairing versus not in a species?
So, yes, Drosophila does something very unusual, and all dipteran insects, well, many dipteran insects, where in cells that are not meant to go on and pair and give rise to egg and sperm, that's where we usually think of pairing as playing a role. Drosophila is unusual in that it does that pairing through almost all tissues, and we recently discovered except their germline during development. And for a long time, people felt that that was specific to insects, dipteran insects, and not seen anywhere else. But over the past 10, 15 years, it's become clear that the X chromosome in mammals, for example, this is work through Jeannie Lee and Edith Hurd primarily, the X chromosomes of mammalian females come together and pair just for a moment at a very critical site. People also have been looking at imprinted loci. There's evidence that there may be some pairing there for the immunoglobulin system, just transient pairing. There is a very interesting cancer called renal oncocytoma, and it's stunning. We're starting to work with that, but this is Kyle Ferg's work. He showed that in cells that are part of renal oncocytoma, they pair their chromosome 19, but only the long arm, not the short, from centromere to telomere. Never seen anything like that outside of Drosophila, not even close. So we are under the impression that actually mammalian cells have the potential to pair and just hold it in check for most of the time.
Okay. Thank you very much, Ting.
Thank you very much.